All right, so this is remediation one, remediation cell cycle. Right. So you were given a task. I said, here you have this cell. You have this cell that has two chromosomes. I'm looking at the nucleus right now. And the two chromosomes, one came from its mother, which had a big A. Mother gave this child a big A, and mother gave this child a big B. Where this creature's father gave this child a small a and a small b. It's a cell, so it looks something like so. There's a nucleus. And I asked you to take it through the cell cycle. One would hope that you would know that the very first thing the cell is going to get triggered on is to go through G1, where it's going to increase in size. The nucleus is not going to change. The nucleus doesn't change, but the cell gets bigger. And that means that if you had endoplasmic reticulum and Golgi body and ribosomes and mitochondria, you doubled or tripled the number of mitochondria. The endoplasmic reticulum doubled or tripled. The Golgi body that doubled or tripled. The ribosomes doubled or tripled. Everything became more and the cell became larger. For those of you that are taking the quiz today, I think that's everybody, you should know that right before you go from G1 to S phase, there is a checkpoint. Arguably the most important checkpoint number one, is right here, right before S phase. If the cell has enough material and it's healthy, it has enough nutrition, and it's big enough, then it goes on and goes into S phase. So those are kind of the things and I'm not writing these things down, but you should have them written down or somehow demonstrated that G1, the cell grows, that the nucleus is the same, and then here in S phase, what you got is basically a copy of what's going on in G1, except now can anybody tell me what's going to happen now? Sorry, what? That's right. Now we're going to get into DNA replication. So now the nucleus is going to, there's something interesting happening in the nucleus. Where in G1, there was nothing really happening in the nucleus. Now, we're going to look at the nucleus. Now, when this doubles, when this DNA doubles, I'm going to have to draw it a little smaller here. Somebody said, well, isn't it going to have an X? And I said, yeah, well, yeah, in a way. What's happening is that these are compacted, they're doubled first, and then they're compacted. And so what you get is something that looks like this when you look under the microscope, and you'll see that today because we're going to have our lab today. And it's doubled, right? So then you have the second one looks like that. And you keep them together so you don't lose them, so they don't get mixed up with a centromere. And since these are sister chromatids, and I would expect those labels, since these are sister chromatids, what I expect to see is that this is a big A, this is a big B, because that's what they got from their mother. This is also big A and big B. These are identical. Sister chromatids are identical. Then again, because they're compacted, they're not unwound the way we had it drawn previously. This chromosome from the other parent is also doubled. It also has a centromere connecting it, keeping it together. And these are also identical, but they're identical to each other. So they're little a, little b. So that is what's going on in S phase. You're just copying it. That's it. That's all that's going on. It's been doubled. The DNA's been doubled. Of course, that would happen in humans with 46 chromosomes. So there'd be 92 copies of the DNA after all this is done and said and done. The next thing is you're going into G2. And in G2, the only thing that's really is going on, you're not really getting that much bigger. You're just checking to see how it all went. You're making sure that there's no errors. If there's errors in the duplication of the material, if there's errors, you just got to check. So what does that suggest that there is in G2? Right before you go into a checkpoint, that's right. Let me make it bigger. I'm going to make it just slightly bigger. It might have gotten a little bigger, but not much. So you have another checkpoint as you go from G2, where you checked everything, to M phase. It might have gotten a little bigger, but in general you have another checkpoint. This is checkpoint 2. Not as critical as checkpoint 1, because at checkpoint 1 either you went through the cell cycle or you didn't. So this really was the probably the most important checkpoint. But here also you can pause. Remember, what are your two options? Pause. The checkpoints have two options. 1, 2, and 3. You either pause, prepare, or die. We call it apoptosis. Okay, so I hate writing with my finger. Apoptosis. So now we're in M phase. M phase is pretty straightforward. I'm just going to draw out, I'm going to do this, I'm going to put a dotted line for the cell. And what am I really focusing on in phase? M phase, what am I focused on? The nucleus. So that's what I'm going to focus on here. I'm going to draw out the nucleus. Well, now we're going to go through the 4 phases. We got prophase, we got metaphase, we got anaphase, we have telophase. And then after mitosis, this is mitosis. Right after mitosis, you have cytokinesis. I'll just put C. In prophase, you have a real condensation of the nuclear of the nuclear material. You have a doubling of the centrioles, which are going to act as anchors for the spindle fibers. The spindle fibers start to form, right? In prophase, and also the nucleus disappears. Now this is important. The centrioles also double, but they also move to opposite sides of the cell. And that allows for the nuclear material to be to be separated. That's going to become important because you need a set of nuclear material on each side of the cell. So then the spindle fibers form. Right? They start to form. The centrioles they've been doubled. They're on opposite sides. The nuclear material's gone. All that happened in prophase. In metaphase, the chromosomes line up. They line up with the sister chromatids facing each opposite direction. The homologs line up. Now, why does that make sense? Why does it make sense that they'd line up in this direction? It's what? Yeah, it's easier to split. You're gonna make sure, because what are the sister chromatids? They're exact copies of one another. So let's remember, you have a big A. You started with a big A from mom and a big B from mom. I think I said that mom. It doesn't matter at this point which who it was from, but. And the other one was a little A from dad and a little B from dad. All right? So you started with, in the very beginning, if we go back to the very beginning of the problem that you were given, you had, what did you have? What was your genotype? Big A, big B. I'm sorry, big A, and big B, little b, correct? Yeah, that's your genotype. So if you're going to end up with an identical cell, you have to have a big A, little a, and a big B, little b. Because you lined them up this way, what is each new cell going to have? Big A, big B, little b. Because this is going to go this way, that's going to go that way. Just because they're lined up the way they are. That would be the drawing of metaphase. Now anaphase, should I draw it? I wonder if I should draw it out, or yes, I should. I was going to just erase, but. So that was prophase of mitosis. Now we can look at metaphase of mitosis. And in metaphase of mitosis, they're lined up. Oh, come on. Metaphase of mitosis, they're lined up. And then in anaphase, what happens? They split, that's right. Now in anaphase, now you're splitting. Now you have one going in that direction. I should draw it this way. One getting pulled in that direction, and one. They're actually pulling themselves with the kinetic core. They have these things called kinetic cores in the middle of the centromere. These kinetic cores are what are eating through the spindle fibers and moving the sister chromatids to opposite sides of the nucleus of the cell. As you can see, they kind of look like they're zooming along. Remember that means that this again is big A from mom and big B from, and this was a sister chromatid, so this is big A and big B, and of course this was little a and little b, and this was little a and little b. So now in anaphase, you can see that they're right. Not just are they separated in number of chromosomes, but they have the right combination of chromosomes in each side of the cell. This is happening in anaphase.
So anaphase when they separate.
When the chromosomes are separating. Now, in telophase, now in telophase they're separated. So I'm going to go ahead and. So they said, oh, come on. So now you have your cell membrane still there, never went away, because if it did, the cell would die. So now in telophase you have this cell, I'm not going to draw the organelles. I'm just going to draw the one chromosome that has the from mom, and the one chromosome from dad, the one chromosome from mom, and the one chromosome from dad. So to, again, to emphasize big A, big B, and big A. Big B, remember each chromosome can have many genes on it, but we're just simplifying it here. Will have many genes on it, all right? So what's going to happen in telophase? You've moved them over. You have a set of centrioles on each side, the way you started. You have the right number of chromosomes on each side. They're the right type. What is going to happen now?
You're gonna have two nuclei, they're gonna form and
That's right, you're going to form a nuclear membrane. That's right. Somebody mumbled something over here as well, but I couldn't hear it, I'm sorry. So the cell membrane stays the same, I just have to expand it because I didn't give myself enough room. So what happens is you start to form a nucleus, two nuclei. So now you got your nucleus is back. But now you have a cell with two nuclei. Right? Two perfect. What is the next step? And still in telophase, this is all happening in telophase. What has to start happening? Cytokinesis. So now you're done with mitosis. Now you have to start doing cytokinesis. So in cytokinesis, you start to pinch off, the cells start to pinch off in animals and other living things without cell walls. They start to pinch off. You still have the nucleus. Let me just try to copy this. Uh-huh. They do, but it's a process. It doesn't just happen, right? So you're right. They're going to end up fully dividing, but first they start to pinch off. They have to pinch off, otherwise you don't. Does that make sense? Yeah. So they start pinching off so that they can fully divide. So they start to pinch off, and then of course, as you just suggested, they pinch off even more. And then finally, what happens? Somebody just said it a minute ago. Finally, they just completely close off from one another, and now you have two cells that are identical. Since you doubled the stuff in G1, they have exactly the same amount of stuff. Are we all good? That would be mitosis, and then meiosis is a little more complicated. I'll record meiosis for you when I do this with tomorrow's kids.
So the cytokinesis have like together.
They're both part of M phase, but mitosis is a division of the nucleus. So M phase includes mitosis and cytokinesis. All right. Are we all good?
Yeah.
So I just gave you at least 100 points there, as long as you wrote it correctly. And if you want the other 100 points, get meiosis straight. Yes, you can say it. You're going to do it all again, but there's differences. Like, what are the differences between mitosis and meiosis? So it does not go through S phase the second time, so you don't copy the DNA again. So when you go the first time in meiosis 1, instead of having them line up this way, they line up as tetrads. All right, if you want to know about meiosis, you come over here. I'll go through it. So otherwise, you can just turn in what you got.
Mr. Mendoza, do you mind, so I wrote mitosis like an example here, but I didn't write it in the cell cycle. So for this, I wrote the cell cycle.
That's fine. Just staple them together. Make sure your name's on it. Make sure everything's labeled and it's clear. If I'm confused, I can't give you points. So
Just go through G1, or is it just big enough?
it's a good question. It's obviously has to duplicate some material.
Where should we start our papers?
Make sure you have your period or class on the paper. I don't want to have to search where you're at. So look, whoever's doing meiosis, if you want to see this, what happens is the big differences in meiosis is you don't do S phase again. You do G1, you grow some things, you make sure you check everything, then you go into M phase. Okay? Yeah, that's right. They're going to divide twice, but you don't want to divide the DNA because then you'll end up, all you're doing is mitosis twice, and that's not good enough. You want to half the DNA. So to half the DNA, you don't synthesize it twice.
-2, sorry.
All right, no problem. So let's take a look at what happens in meiosis. So in meiosis 1, in meiosis 1, instead of the lining up during mitosis of meiosis 1, during prophase, first of all, I should say, in meiosis 1, you have what's called a tetrad forming. Tetra means 4. So what that means is, if we look at it, what that means is that you have the little a and the little b, and the big A and the big B. Just put it there. You don't need to talk to me. You have a tetrad forming in prophase 1. So one of the big things that happens in prophase 1 is that there's a switching of DNA. Randomly, a piece of these two chromosomes will switch. So what you'll get is some of mom's DNA being mixed with some of dad's DNA. So instead of big A, little a, big B, you have little a, big B here. And on this one, instead of little a, little b, you have a big B here.
Wait, okay,
So now you have 4 different combinations. You have big A, ooh, I put big A there twice, that's a mistake. You have big A, big B, which was original. You have little a, big B, that's the mixing up. The crossing over happened, that's what crossing over. You have big A, little b, right? And little a, little b. So you literally mixed up the DNA. That's why we call it recombination.
so I see that there's 4 of them. Does that mean you're separating the sister chromatids?
Excellent question. No. So in mitosis, you separated the sister chromatids.
Yeah.
These questions would be great in class. Will be great in class, should be great in class. Ask them in class, okay? In mitosis, you separate the chromatids, sister chromatids. In meiosis, you're separating the homologs. So these are staying together. So now during M phase, or during metaphase of M phase, right? During metaphase of meiosis 1, so metaphase 1, what happens is that the centrioles still divide, they still separate, spindle fibers still form, you still get kinetic cores, but now these things, instead of separating the sister chromatids, now you get,
Is it time for class?
I don't know. Now you get this chromosome, this homolog. I shouldn't do green. Let's do red. This homolog moving one direction, and this homolog moving in the other direction. So you get this one moving this direction and this one moving in that direction. So they separate as a pair. Does that make sense?
Are there names for these cycles, or is it just called meiosis one?
Meiosis 1. And then metaphase 1. This is metaphase 1, and then anaphase 1. And then telophase, after telophase and after the first cytokinetic event, what you have are two cells, right? Think about what you're going to end up with because you did this. If you think about what you end up with, there's no doubt you know what you're talking about. Come on. At the end of the first cytokinesis, what you get is this pair of chromosomes that went through crossing over, correct? So you have one set that's still big A, big B, one chromosome still big A, big B. Then you have another chromosome, though, that's big A, but then little b. And you have another one that's over here. Oh, I'm sorry, I messed that up, didn't I? Yeah, that's right. I didn't do that before. I think I did little a over here.
Do they still have checkpoints again?
Yeah, you still got checkpoints. That's still happening. You have little a, big B, and over here you have big A, big B, and over here you have a big A, and then you have the little b. Does that make sense?
Yeah.
I messed that up. Does not make sense. This should be little a, little b. I don't know what you're doing. Whatever you're doing, just do it. I have to finish this thought. So what you end up is big A, big B, little a, big B, little a, little b, and big A, little b. So you have 4 different options. Now you do mitosis. Meiosis 2 is just mitosis. Now instead of lining up there's no tetrads, no crossing over in meiosis 2. They line up on the metaphase plate just like they did in mitosis. But now you only have half the DNA because you didn't do S phase. Do you remember that?
Yeah.
So because you did half the DNA, now you end up with 4 cells, right? Because each one of these is going to divide, right?
Yeah, so after meiosis, then they go through mitosis again.
Well, no. No, they just do meiosis again. But no, it's just like mitosis. Meiosis 2 is just like mitosis. But there's no double of the DNA. So now you have one gamete, these are sex cells, with big A, big B. That's the original form, right? Shush of one of the parents. One that has little a and a big B. And one that has a little a, little b, and one that has a big A, little b. So now you have this recombination, right? This different set of DNA. Instead of 2 different options, big A, big B, little b, you got 4 options. That's why you have a Punnett square the way you do. That's why there's 4 spots in a Punnett square, because there's 4 possible combinations in between 2 heterozygotes. Does that make sense?
Yeah. Do you
All right.
want, is it okay if I finish this? Just like finish writing down what you said, or just want me to
No,
turn it?
just turn it. Whatever you did. All right?
I have to staple.
Yeah, staple it and go. All right.
Was it here?
Huh?
Here?
Yeah. So that is, that was half an hour, perfect timing. This and say, what we're talking about here is mitosis and meiosis. And with mitosis and meiosis, there's 2 processes. They're very similar. They both have to do with the nucleus. That's their focus. And the nucleus, though, the mitosis and meiosis are part of a cell cycle that starts with G1, moves on to G2, moves on to, oh, not G2. No one's corrected me. Why is that? Thank you, it's S, thank you. It's G1, S, and G2, and then it goes on to M phase, in this case, mitosis or meiosis. And then, of course, as part of M phase, is cytokinesis, but it's not part of mitosis and meiosis. Cytokinesis. So there's two parts to M phase. Part one is either mitosis, meiosis, then part two is cytokinesis. Then you either go to G1 or you go to G0. And G0 is just where you do your job as a cell, whatever that job is. I want to remind you the cell cycle is about dividing the cell division. Also, I want to remind you that this is only done in eukaryotes because you have an M phase. Prokaryotes do cell division. So this is eukaryotes. Prokaryotes do cell division as well. They just do binary fission. Their cell cycle is their steps to binary fission is nothing like the cell cycle. Well, not as complex as the cell cycle, let me put it that way. And the reason is they do have to copy the DNA, but they don't have as much DNA to copy. They do have to duplicate their organelles, and they have to increase their size, but they don't have any internal membrane bound organelles, right? They don't have any of that. So they don't have they can do binary fission, cell division, in other words, very quickly. They don't have to do all the steps that eukaryotes do, etc. But they do have to replicate their DNA. They do have to check for mistakes. They do have to they do have to make more of themselves in order for them to be able to divide. So none of this should be new. None of this should be new. Should be all pretty, again, if it's new to you, then they have to ask yourself why. All right. So that's the concept there of cell division. Now the difference between, let's go over meiosis and let's try to remember what mitosis is. We did a lab. Did you guys do the lab? I can't remember if you did the lab or not. Yeah, you did. The Friday kids weren't able to do the lab. They did you saw there was a virtual lab? All right, so if you wanted to do the virtual lab, I think the second virtual lab I posted works. The people do it. Anybody in here do the virtual lab? I changed the virtual lab. The first one had issues. It went on. It worked, but it did some odd behavior. So I did another virtual lab. Yeah.
Is it the one that you worked with me on, or is it just the one with the pictures?
It's a. I can't remember. I don't know if I worked with you on it or not. But there were if you do the lab, there's a bunch of slides. You shouldn't have any cell phones on, right? That's a good thing about a dark room. If I see your light, so you need to have your phone off. And I understand you're leaving soon, but regardless, you had your phone on, right?
Yeah.
Your computer. Thank you. So the bottom line is, when we're talking about that lab, it does give you practice of the steps of mitosis and what it looks like under a microscope. It looks a little bit confusing, but that's what it looks like under a microscope. As you saw, it was confusing. And what the pictures you see It's not as nice as the things I draw, right? And a lot of people, some kids are like, well, why are you drawing? I can't learn from these drawings. I need hands-on. I give you hands-on, and you're not, as you saw, hands-on is confusing because the real world is a lot messier than the simple drawings that I draw on the board. All right. So let's go on and look at meiosis. So in meiosis, think about the big picture. Always, I try to give you stories and try to give you big picture ideas. You have two people. I'm using people because they help you to help you relate. But you can use. You have two whales. You have two. Whatever. I'm going to go ahead and use some gender to designate female, male because I can't draw. So that's what I had to. So. Male, you're going to try to make a new person. This person has 46 chromosomes. This person has 46 chromosomes. Now, that statement is a statement that a lot of you will remember. But then you have to remember there's 20. Each of these people have 23 chromosomes. Twice. They had 23 pairs. They got 23 from their mom and 23 from their dad. That means half. So they got chromosome number 1 and 2 and 3 and 4 and 5 and 6 and 7 and 8 and 9 and 10 and 11 and 12 and 13 and 14 and 15 and 16 and 17 and 18 and 19 and 20 and 21 and 22 and 23. The 23rd chromosome is a sex chromosome. And if it came from mom, it's an X. Only one of each. This woman got one of each. Is everybody okay with that? From their mother. Their dad gave them the same thing. Exactly the same thing. 22 chromosomes. And he also gave her an X. There's a reason I'm doing this. There's a reason I'm telling you to look at this and think about it. Take a breath and think about what they're saying because there's implications to this. There's sex-linked diseases like there's trisomy 21. There's chromosomal abnormalities, triploidies, etc. There's all kinds of genetic diseases and genetic issues that come from understanding that you get 23, half your DNA from mother, half your DNA from father. That objectively is easy, but there are implications to it that are immense. Yes.
So, does the dad have a random chance to give the
Yes.
X or Y?
That's correct. Because remember, if we take a look at mom and dad, let's take a look at mom and dad. This woman's mom and dad really quickly. This dad has this set of chromosomes, and he has another set of chromosomes, except he doesn't have an X. He doesn't have two X's. What does he have? He has an X and a Y. Oops, shoot. Okay, I can't undo that. That's really 20, 22, and he has a Y. So he has. So the question is, is it a 50-50 shot that this dad gives an X to his daughter versus a Y? Yes, absolutely. It in fact, I don't know if you know about Henry VIII. Yeah. Yeah. Henry VIII wanted to have a son. He needed to have a son. That's the way these royals do, right? They have to have a son, an inheritor, etc., etc. The Roman Catholic Church would not give him a divorce. All very political, very ugly, icky stuff. And so he decided to come up with a pretext and kill his wife. He killed a bunch of them. I don't know all the details. You can watch the shows and look at the history. I'm not a history teacher, but he killed his wives because the theory is it was the woman's fault. Everything was the woman's fault. Everything. The guy can't be the king, can't be the guy. It's the woman's fault. That's sarcasm in case you're missing it. But so that being said, it wasn't her fault at all. It was him. She didn't give him anything. They had a bunch of young women, female babies. He's the one who was providing the eggs. Maybe he should have, there's actually, it's a little more complicated than that, but still. So this person's mom, this female had a mom and a dad, and they had a mom. Their mom had 2 exes. Their dad had an X and a Y, and it was this X that got given to this Y, to this mother here. All right. Which one of these 2 X's did this woman get? I don't know. It's random. So which set of all 23 of these are random. Independent assortment. 23 chromosomes with genes on each one randomly. Which chromosome do you get? The mother, the father? We don't know. It's a random event. And we'll see how random it is in a minute. So in this case, this woman got an X from her dad. This man got a what from his dad? A Y. Same situation you see here happened with the man, except he got a Y. All right. Well, let's look at how they did that. What that meant that we had to take these 46 chromosomes and divide them. Now, it can't be random. It can't just be like bingo balls, right? And pull out number 20, because you can pull out a number 1, and you can't just divide it evenly, take half the bingo balls and move them here, and half take a big knife and separate in half. You can't do that, because then you could randomly get 23, 2 number 1s and no number 2s. Right? And the other person get 2 number 2s and no number 1s. That's no good, because number 1 might have the gene for lungs. Oops, that doesn't work. Ooh, no brain gene or some essential gene that you need to survive. So that can't happen. So there has to be an organized, very ordered way of doing it. And life has evolved this really simple solution, and it's a beautiful thing. It's called meiosis. So, and by the way, why do we have to divide it in half? Why do we have to divide it in half into what's called gametes? Why do we have to make gametes at all? We have a sperm in humans, a sperm and an egg. Why do we have to have sperm and eggs at all? Yeah. That's right. So the chromosome number stays at 46. If we have too many chromosomes of any one chromosome, it's called a trisomy or the tetrasomy or whatever. It's bad. Bad things happen. What happens? It depends on which chromosome. 3 copies of chromosome number 21 gives you Down syndrome. That's not a great thing to get. I mean, I've known children. There was a kid in my church group that had Down syndrome. She was a great lady. Fantastic. But she has to struggle through a lot of stuff that she wouldn't normally have to struggle through. So having too many copies is no good. It doesn't work. You have to have a regulated number of copies. So you get the right concentration of proteins that do the right function. Too many of any enzyme, you get things going on that are out of control. So meiosis helps us do that. We have to. So what do I mean by that? Let me make it simple. If we had 46 and we combined our genes with another person who had 46, we'd have 92. If we had 92 and we married somebody and they got together and had a baby, we'd have 184, and it would keep going. It sounds like it's getting out of control. It already got out of control here. This person was never born because it just couldn't survive. It's producing too many gene products that are going to hurt the process of life. So meiosis, you start the first step with G1. I'm going to go ahead and start and say we've already copied the genes. And I'm going to do this with one cell. By the way, this was the remediation. The remediation was to take this sample cell that I'm just going to use. I'm just going to use one gene, one chromosome. This cell is not human, only has one chromosome. And it's going to go through meiosis. So when we're looking at this, you go ahead and you take, I'm just going to do meiosis. You'll do mitosis as well. So you have a single chromosome from their mom, has an A, and here has a big B, and here's a single chromosome from their other parent that has a little a and a little b. Now, in this example, it's big A, they got a big B from mom and a, little b from their dad or whatever. Everybody understand that? Okay. You take this cell through both meiosis and mitosis and accurately, and I'll give you your 400 points back on your quiz. That's your remediation.
Take what?
Take this cell with just this one chromosome, right? Only one homologous chromosome. By the way, let's get our vocabulary straight. This one and this one are homologous chromosomes. They have the same genes, right? They both have A and B on them. Does that make sense? So they're homologous. Are they the same? Are they identical? What's different? Little a and big A and little b and big B. So what do we call these homologous? What do we call those big A and little a? Alleles. So they have different alleles, but they have the same genes. So we call this homologous chromosomes. So you take this one single pretend cell, it's a made up cell with only one chromosome, one pair of homologous chromosomes. It's a diploid organism. The word is diploid, which means there's two copies of every chromosome. You're diploid, dogs are diploid, this pretended creature is diploid, okay? Take this and do mitosis and meiosis with it. I'm going to do meiosis with you right now. You get half of it done right now. Do the other mitosis. Do mitosis on your own with it. And I'll even point out the differences between meiosis and mitosis. Well, what's the first thing that this cell is going to do if it's going to go through meiosis? What's it going to do?
It's gonna like turn into the three thing. Like it's switch over to.
Well, think it's a cell. It has that nucleus, right? What is this cell? What is the first thing it's going to have to do if it's going to go through meiosis? That's right. It has to double everything. So what do we call that? What do we call? What phase is that? G1. That's right. So it has to go through G1, doubles everything except the DNA, right? So it gets bigger. Doubles everything except the DNA, gets bigger. Then before it goes on to what phase does it go next? That's right. And what's in between G1 and S? Checkpoint is correct. Checkpoint 1 is here, where it's going to check for the size of the cell, make sure it's big enough, make sure everything's been doubled. It's not checking DNA at this point. Then in S phase, it's going to do DNA replication, correct? And then what does that mean? What do we end up with in S phase, at the end of S phase? right? We end up with what? Two sets. It says two individual homologous chromosomes, right? There's still homologous chromosomes, but we end up with two sets. Does everybody understand that? So we have this nucleus, we have this set of chromosomes here. They're not condensed, they're just copied. They're connected and copied, but they're not condensed. You wouldn't see them like this at this stage. You'd see them like this at prophase, in the beginning of prophase 1. But we're going to go ahead and draw them out like this here, just so that you can clearly see what I'm saying. There's two sets of them, and they're identical. So this is big A, and this is big A, and this is big B, and this is big B. In fact, if you don't mind, I'm going to go ahead and do different colors and just go ahead and do this one in red just to keep it clear that there's two different the homolog pair are different. And over here you have little a and little a, and here you have little b and little b. So there's two copies of every chromosome. So we've doubled the DNA. That's called S phase, synthesis phase. Then it goes on. What happens as it goes on then to what? G2. And what happens in G2? What's going on? Is there anything between S phase and G2? Is there a checkpoint between S phase and G2? So yeah, there isn't. So that's interesting. Is there one during S phase? No. So that's interesting. Now, to be clear, when it's going through DNA synthesis, when you're copying the DNA, it is checking for mistakes and correcting. But there's no checkpoint. It checks and corrects. There's no decision point where it says apoptosis, the cell dies, or the correction. It just corrects. That's what we mean by checkpoint. All right. Okay, so then it goes on to the next. This is G2. So you have this thing. It looks like this in G2. Now we're into M phase. Correct? Now there's going to be a point in M phase where we end up with two cells. All right, that's called cytokinesis. That's part of M phase. But now we're going to look at meiosis. And in meiosis, which is part of M phase, you have prophase. And in prophase 1, why do I say prophase 1? Because what? There's more than one. That's right. I say prophase 1 because there's actually two steps to this, two series, two changes. So it's not just one. All right. So in prophase 1, now this would be the same. You do the same doubling in mitosis and meiosis. The difference is that in prophase 1, instead of being separate like this, what happens? Does anybody know what happens? They condense, but they don't just stay separate, two sets of sister chromatids. What do they become a tetrad. So they become linked. They become linked together. So there's not two homologs. The two homolog sets of the homologs are copied, are stuck to each other. They form a tetrad. Tetra means 4. So 1, 2, 3, 4. The 4 sister chromatids are together. So that's happening in prophase 1. So they're already stuck together. And then the other thing that happens in prophase 1 So they form a tetrad. And then 2 is they do crossing over. All right, so they cross over. What does that mean? That means wherever these chromosomes touch, the sister chromatids touch, they break and switch their sequences. So what happens is this breaks off and you get the little a over here, and over here you get the big A. So now you have big A, big B, but then you have little a, big B, and you have big A, little b, and little a, little b. Before you only had 2 different copies of 2 different options, big A, big B, or little a, little b. That's the only options you had. Now you have 4 different options. 4. Big A, big B, little a, little b, big A, little b, and little a, little b. Very important. So let's say big A means tall and big B means yellow, flower. So one could have made a tall yellow flower if you had 2 copies of it. The other would have made a short red flower, no, short yellow flower. Another one would have been a tall red flower, and the last one would have been a short red flower. You'd have 4 different kinds of beasts, where before you mixed up the DNA, that's called, by the way, the word here is recombination. You are recombining the DNA. You're increasing genetic diversity / variation. You've just increased genetic variation. You went from 2 possible to 4 possible combinations just by crossing over. And it's random, by the way. We don't know where the crossing over is going to be. Now, the other interesting thing is that in metaphase, because that's the next step. Now, by the way, in prophase 1, don't forget that the nucleus dissolves, the nuclear envelope dissolves, so there's no so now you can move them, now you can separate them. Don't forget that the centrioles also doubled and moved to opposite sides of the cell, and that the spindle fibers started to form. All right, all that happened in prophase 1. So now in metaphase, which is the next step, correct? Metaphase 1. Now you have your centrioles. You have your spindle fibers, but now your tetrad, instead of the homologs lining up one on top of the other, now you have a tetrad lined up. How does that change things? Think about it and try to figure it out on your own I have not told you this. Yeah, I did in 9th grade, many of you, but you probably don't remember. But now the spindle fibers, instead of connecting, lining up the homologs one on top of the other, because remember, if we in mitosis, guys, if you're gonna have the side conversation, go outside, please. Just go. I'm okay with it. Just go outside. To sit there in the middle of this conversation and have a conversation is so insulting and disruptive. So now in mitosis, what happens is they line up this way. Now that's a difference. Is it not a difference? And so there are consequences to these differences. What is the consequences? What can you see as a consequence? What are the kinetic cores in the center of these centromeres? What are they going to do in mitosis during anaphase? What's going to happen to these two? They're going to pull apart, correct. So in anaphase, this one goes this way, that one goes that way. This one, now these are homologs, they're lined up on top of each other. So what happens is that this one has a big A, and big B, where this one has a little a, little b, little a, little b. In mitosis, at the end, during telophase, you'll end up with one big A, one big B, one little a, one little b in each of the new cells, correct? All right. So that's mitosis. But in meiosis here, what's separating? The tetrad separating, correct. So remember, that's little a, little b, little b. This one, little a, is moved over here because they're crossing over. Here, this one has a big A over here because it's crossing over, and this one's big A and big B. So now you're gonna end up separating those two. Now those two are going to separate, and what you're going to end up in telophase is you're going to end up with a cell. Let me erase M-phase here. So in telophase 1, you end up, and these have been questions that have come on tests. So I'm telling you right now that this is very testable. In telophase 1, you end up with one homolog over here, again, the same genes, and one homolog over here. The sister chromatids are still attached. So you only have one of each of the chromosomes here. This one has a big A, this one has a big B, this is a little a, and this one's a big B. Where that one over here is a little a, this is little b, this is little b, and this one's a big A. I skipped anaphase. Anaphase happened in there too, yes. There is no, again, the only difference is that in anaphase 1, the homologs separate, right? The tetrad separates, not the sister chromatids. So then you end up at the end of meiosis 1, you end up with two cells. At the end of M phase 1, you end up with two cells, both with a set of chromosomes that are doubled sister chromatids, one big A, one big B, one little a, one big B. And the same over here, you're going to have, because you separated the homologs. So now you have these two cells. What are you going to do with those? You're going to do what? The same thing. You're going to do meiosis again. You're going to do the same thing. You just have the DNA, but you still have, technically, you still have the same double, right? So here's the wording, because Chardon said it correctly. It's all about wording. So if they ask you at this stage, at the end of meiosis 1, is it true that you have two copies of two chromosomes in this diploid organism? Look at it. At the end of telophase, at telophase 1 of meiosis, at telophase 1, which is after the first division, after the first meiotic event, how many chromosomes do you have in each cell?
2.
Trick question.
4.
1. in this case.
Wait, cause they still like.
They're still connected.
Oh.
You have, now, if you asked you how many copies of each chromosome, of each of the DNA sequences, you have 2 copies. If they ask you how many chromatids, that's 2 in each new nuclei. But 4 total in the cell, because they're still together. It's only after cytokinesis are they separated. So Chardon's absolutely right. It's all about wording. How do you word the question? It's not that you don't know. You know how it works in general, but they're going to get specific. They're going to ask you a question, and they're going to say, wait a minute, how many chromosomes are here? How many copies of the DNA are there? Is this creature still diploid during telophase 1? Technically not. Technically, after the first division, they're already haploid, but there are still 2 copies of the DNA. There's still 2 sister chromatids. So what do we have to do? Because we don't want all these extra copies of the genes. What do we need to do? We need to divide them again. Do we go through S phase? Should we go through S phase again? No, why not? We have everything. We don't want to double the DNA. If we double the DNA, we'd end up right back where we started from. Except we'd be mixed up. We have to keep it because we want half. So each of these are going to divide into 2. It's going to do, meiosis is going to be meiosis 2 is almost exactly like mitosis. In fact, there's no difference except there's half the DNA. They line up, as we looked here, they line up the DNA, so the sister chromatids can be separated. That's it. And then you end up with these. Each of these is going to divide into 2. You're going to have one cell here with one nucleus, with one single copy, one chromosome, haploid, half the DNA. That one will have the tall gene. That one will also have the I don't know, whatever the color I said, yellow gene, right? You'll have another cell, another sperm or egg, depending on whether it's going through oogenesis or it's going through spermatogenesis, whether you're going and making a sperm or an egg. You have only one copy, so it's haploid, half the DNA. And this one has one big B, so it's red instead of yellow, but it'll be short if it met up with a similar combination of its opposite gamete. Then it'll have another cell, another egg or sperm that'll have a nucleus that has, it'll be tall, just like the other one was, but then it'll be the red instead of yellow. Again, assuming that the genetics works out, because it all depends on is it dominant or recessive. And so this is the big A, and then it has a little b. And then there'll be another cell, egg or sperm, with yet another nucleus, except this one's going to be both short, have the gene for the short and the red. So this has the gene that could make a short and could make a red, depending on what the other parent gives. This one has the trait that could be the genes that could lead to tall and could lead to yellow. This one has the trait that could lead to short or could lead to red. And this one has the trait, the genes that could lead to the trait for tall and for yellow. So you see, this, what used to only give you two possibilities, now can give you 4 possibilities. And then these go on to develop into either eggs or sperm. This is meiosis 2. there is no very little difference between meiosis 2 and mitosis. It's only in meiosis 1, in prophase 1, that you form a tetrad versus lining them up in a straight line. It's only in meiosis 1 that you have crossing over. You don't have any of that happening in meiosis 2. It's only in meiosis 1 that you separate the tetrads instead of separating the sister chromatids. In mitosis, you separate the sister chromatids during anaphase. In meiosis 2, you separate the chromatids, just like in mitosis. Yes.
Like, so is big B supposed to be yellow tree?
Yeah. I should like. So if
Short yellow?
so, big A would give you the trait for tall. Little a would give you the trait for short. Assuming you have what? 2 little a's, right? So you have. Does that make sense? Assuming this is a dominant recessive. This is only these are possibilities. Then the little a would give you. I'm sorry, not little a. Big B would give you yellow, and little b would give you the possibility for red if it's dominant recessive. If I have bigger and little, it suggests dominant recessive. So in a dominant recessive world, inheritance, it all depends on what the other parent gives you. So now you got all these different combinations, and that's just for 2 genes with 2 alleles. Can you imagine what happens when you have 10,000 genes with 3, 4 possible alleles in each gene? The crazy variation that we call life happens. So it's no wonder that you can get a single living cell turning into all these possibilities that you see around you, all this life that surrounds you. So yeah, so anyways, that's a cell cycle, that's meiosis. Your job, if you want to remediate last week's work, is to draw this out for mitosis and meiosis. I've drawn out most of meiosis 1. You'd have to draw out meiosis 2, which is just like mitosis, except you're only dealing with half the DNA. This is the kind of work that some of the, like this was a 9th grader who did it, so this is the kind of, this I think gets you 100%. See what she did, just drew it out. Alright. That was meiosis 1, that's meiosis 2. A lot of the kids there, some of the kids wanted to, anyways, that's it. Alright, so are we good? When's the class over?
45.
At 45 minutes after, is that right? Or 48?
It's 48.
48, okay, so we still have a few minutes. You can start now if you want to do it. Do I have any questions? I don't want to start a new topic. Tomorrow we'll finish the checkpoint discussion and make sure you make sure we get the wording straight. That's very useful, Chardon, when you say stuff like that. That's very useful. I only have 5 staplers in here.
You only have 5 what?
I only have 5 staplers in here with several boxes of staples.
Oh.
Did you want to turn it online?
The what?
You said you were going to post an assignment online.
Yeah, I will. I'll post it online. You can just upload it.
Because I came in, but I ran out of time and couldn't do meals.
That's fine. I don't care, as long as it gets done.
Are you sure? Or should I leave like 2 minutes early?
How about I let you all out 2 minutes early? I let you. See, this is. I feel a conspiracy is afoot. They're whispering. It's just shaking hands and hand. It's like you and him. He's at the center of all conspiracies in this room, and then he does it with a smile. That innocent face.
I'm doing nothing.
Yes. No, I wouldn't go that far. I would say that all the differences between meiosis and mitosis are in meiosis. It's just doing like mitosis. You're dividing it. It's important to divide the DNA. You have to separate the DNA. It's important. You need 4 cells that are half the DNA each. That's part 1. The other part of meiosis is to mix up the DNA. That happens in prophase and meiosis 1.